In this chapter, we will study thermodynamics, specifically the thermodynamic values of a cell. Delta G, delta H, and delta S. For the experiment, we will use electrochemical cells, specifically galvanic cells, which consist of 3 electrodes. The first electrode, copper, acts as the cathode, positive, and the second electrode, zinc, acts as the anode, negative. The key components of the cell are the electrolytes. For copper, the electrolyte is copper nitrate, and for zinc, the electrolyte is zinc nitrate. An important element is the salt bridge. Our salt bridge is made from a stack of paper, cut into long strips, and immersed in saturated potassium nitrate. Next, we will assemble the cell, which is a glass tube. There's a connection between 2 glass tubes. With each tube serving as 1 electrode, we'll have 2 electrodes in total, 1 copper and 1 zinc. To set it up, we pour the electrolyte solution. The blue solution is copper nitrate into the tubes. It will flow through the central opening. For the second electrode, we'll pour the zinc nitrate solution. The clear, colorless solution is zinc. We'll add the zinc to this solution. For the blue solution, we'll add the copper. To connect the cell, we'll immerse the 2 glass tubes in a water bath. Because we'll be heating them, we'll then immerse the copper electrode in the blue solution and the zinc electrode in the colorless solution. The remaining step is to insert the salt bridge, which is a long piece of paper. We might need a little help inserting it. And we'll need to check that the salt bridge is properly submerged in the solution. Once the cell is connected, we're ready. Next, we will read the electromotive force, or EMF, while simultaneously recording the temperature. We will now immerse the thermometer in the water bath. Once the thermometer is submerged, we need to read the temperature at that moment, and then press the button on the device to read the EMF value. The result will be 0.8892. This is the EMF value at this temperature, which today is approximately 30.2 degrees Celsius. Therefore, we will continue reading the EMF values as the temperature increases by 5 degrees Celsius. So, starting at 30 degrees, then 35, 40, 45, and 50 degrees. We will then turn on the heater. At the same time, we need to activate the magnetic bar. As we calculate, the electromotive force, EMF, will decrease as the temperature increases. We will wait until we reach our desired temperature. Then we read the EMF values, doing this until it reaches 50 degrees. Then we stop the heating by turning off the heater. Now, we'll also turn off the magnetic stirrer, because we need to lift the beaker to prevent it from still being heated by the heater below. If it's not too hot, one student can help lift it, and another can place a tile to protect the cell from the heater heat. Once that's done, we'll turn the stirrer back on without doing anything to the heater because we're going to cool down. Therefore, we'll use ice for cooling. We want it to cool down slowly, so we can read the EMF values in time. We'll add the ice gradually, 2 or 3 pieces at a time, and observe the temperature and how the air circulates. Reach the desired temperature. That was the starting point, 50 degrees. We'll read it. As it cools down to 45 degrees, we'll repeat this process until the temperature reaches 30 degrees again. That completes the first part. We need to do this experiment twice. In the second part, we need to change the solution inside. We need to replace everything, including the salt bridge. The electrodes will be washed with distilled water and dried with tissue before being used a second time. Everything will be done the same way twice. Then use the experimental results to calculate various parameters.